Cell cycle regulation is the complex system that controls when and how cells divide. The cell cycle consists of four distinct phases: G1, S, G2, and M. In G1 phase, the cell grows and prepares for DNA synthesis. During S phase, the cell replicates its DNA. In G2 phase, the cell prepares for division by producing proteins and organelles. Finally, in M phase or mitosis, the cell divides into two daughter cells. The transition between phases is controlled by checkpoints that ensure conditions are optimal for division. The G1S checkpoint ensures the cell has grown large enough and has undamaged DNA before proceeding to DNA synthesis. The G2M checkpoint confirms that DNA replication is complete and accurate before allowing the cell to enter mitosis. The metaphase checkpoint verifies that all chromosomes are properly attached to the spindle and aligned before cell division proceeds. Key regulatory proteins called cyclins and cyclin-dependent kinases act as molecular switches controlling progression through the cell cycle. Cyclin concentrations rise and fall throughout the cell cycle, triggering progression through different phases. While cyclin levels fluctuate, cyclin-dependent kinases, or CDKs, remain at constant levels. When cyclins bind to CDKs, they activate these enzymes, which then phosphorylate target proteins to advance the cell cycle. External growth factors from neighboring cells also play a crucial role in signaling when division is appropriate. These growth factors bind to receptors on the cell surface, initiating internal signaling cascades that ultimately affect the cell cycle machinery. To summarize, cell cycle regulation involves distinct phases, critical checkpoints, regulatory proteins like cyclins and CDKs, and external signaling through growth factors. This complex regulatory system ensures that cells divide only when conditions are favorable, maintaining the balance needed for healthy tissue growth. When the precise control of cell division fails, serious consequences can result. If cells divide too frequently or without proper regulation, cancers can develop, essentially diseases of uncontrolled cell division. Conversely, if cells fail to divide when needed, tissues cannot be properly maintained or repaired, leading to degenerative conditions. The body maintains this delicate balance through multiple redundant systems. Tumor suppressor genes like P53 act as emergency brakes, halting division when DNA damage is detected. Contact inhibition prevents cells from dividing when they're surrounded by other cells, maintaining proper tissue architecture. Telomeres, the protective caps on chromosomes, shorten with each division until they reach a critical length, triggering cellular senescence, a natural limit on cell divisions. This prevents potential mutations from accumulating over time. The multilayered system ensures that cells divide exactly when needed to maintain tissue health.